to Medical Grand Rounds. Before we introduce our speaker today, just a reminder that please let us know that you are present at rounds today by answering a brief survey that will be sent to you right after Medical Grand Rounds. And please keep your questions to the end.
Okay, thanks, Nadia. So it's a pleasure today to welcome
and enter your question so that we can ask.
You're cutting out.
So actually, sorry about that, George. I don't know if you can hear me. I'd like to ask George Fantis to present our speaker today, whose speaker is coming on behalf of the
Okay, so Nadia is cutting out a bit, but I'd like to welcome everybody to Medical Grand Rounds today. And it's a pleasure to introduce one of our members of our endocrine division here at McGill, Brent Richards. Brent is a professor at McGill and one of our Department of Medicine and Division of Endocrinology members. He's cross-appointed to epidemiology as well as medical genetics. Just a brief background, he went to undergraduate at McGill in genetics and worked with none other than Charles Scriver. For those of you who don't know, Dr. Scriver is now 91, and he was a fantastic mentor, obviously impassioned Brent to follow in his footsteps in genetics. Charlie was one of the sort of founders of modern genetics. He wrote a book on the inherited basis of metabolic diseases, introduced testing for phenylketonuria, solved the problem of vitamin D and rickets, and helped to introduce vitamin D into milk, amongst other achievements. Brent went on to medical school in Calgary, followed by residency training at McGill and an MSc in epidemiology, working with Dr. Goldsman here in osteoporosis and vitamin D. He then went on to King's College in London, St. Thomas Hospital to do genetic epidemiology, and returned to the Lady Davis and Jewish General Hospital and our Division of Endocrinology. His work is noted on genetics and endocrine disease, particularly osteoporosis and vitamin D. And he's actually linked vitamin D levels to multiple sclerosis. He's won many awards for his work, 2 Young Investigator Awards, one from the Canadian Society for Clinical Investigation and also from the Canadian Society of Endocrinology and Metabolism. He's been elected to the Royal Society of Canada College of New Scholars, Artists, and Scientists, and he's also elected to the prestigious American Society for Clinical Investigation. His work is very high impact with a very high index of citations. And today he's going to show us some research where he's pivoted from endocrinology and genetics into genetics and something that's on our minds almost daily for the last 2 years, COVID. So welcome, Brent. We're looking forward to your talk.
Thank you so much, George, and thank you, Nadia. And indeed, I really owe a great debt to Charlie Scriver, who was a mentor early on in my career, but continued to be so, and really excited me about human genetics, probably at the right time. So I'm going to talk about the influence of OAS1 on COVID-19 outcomes and Neanderthals to the rescue. So I'll start with my conflict of interest slide. And the most important thing that you need to understand is I've probably been biased by the fact that I have more Neanderthal DNA than 94% of other customers of 23andMe. You probably already knew that, but that is what my genes tell me. And I also should declare that I have recently founded a small spin-out from our lab called 5' Sciences. So all this work is, of course, not done by me. It's predominantly done by these 3 people. So this is Suriju. So she just started as an assistant professor at the Genome Center. She's a human geneticist who led this program along with Guillaume Butler-Laporte, who is an infectious disease specialist and is doing training in our lab and shortly be off to Oxford, and hopefully will return back to the McGill fold. And Tomoko Nakanishi, who is a MD and respirologist from Japan, who joined us to do her PhD in human genetic research. And so, as George pointed out, I'm an endocrinologist and of course know nothing about infectious disease, but for reasons which I'll explain in a moment, we got caught in an opportunity to try and make a contribution to understanding the genetics of COVID-19 outcomes. And really, I felt very well supported by having a human geneticist, an infectious disease specialist, and a respirologist leading this program. Lots of other people contributed to this, and I'll just briefly mention them at the end. So the reason why we got caught up in the maelstrom of COVID-19 research is because we were in the midst of creating an institutional biobank at the Jewish. And when this happened, the FRQS approached Vincent Musser, who, as you may know, is the Canada Excellence Research Chair of Human Genetics at the Genome Center, and asked him to create a province-wide biobank to collect data and samples for people in controls who had experienced COVID-19. This led to what is called the Biobanque Québécoise de la COVID-19. This is the largest collection of data and samples for people who are predominantly hospitalized with COVID-19 and their controls in Canada. It's amongst the largest in the world. This is an open science biobank. And so you can apply with very few hurdles to access the data. You just have to have ethics permission and agree to treat the data within the provisions of its confidentiality, as well as pay a small access fee. This data set is quite rich. So we have 3,431 participants. They've been recruited over the pandemic. About 2,400 of them have severe COVID, and there are about 1,000 that have mild COVID. We have a lot of samples, and these have been collected across 8,279 visits. These include a whole bunch of different samples which you can apply for to get access to, including PBMCs, serum, DNA, RNA, etc. And really, by the grace of investments from the FRQS, the Public Health Agency of Canada, as well as Genome Quebec, we've been able to undertake a fair bit of analyses on these individuals. So this is a set of 1,000 patients with clinical and phenotypic data, so a subset of the cohort from 2,000 visits, wherein we've undertaken proteomic analyses, whole genome sequencing and GWAS, transcriptomics, multiple immunology, serology panels, metabolomics, standard of care laboratory measures, as well as standardized questionnaires and follow-up that is done on all of their visits. So this is a very rich data set. We encourage you to have access to it. Almost everything that I'm going to show you today was derived from that data set, and I'm sure many of you as clinicians and members of the Department of Medicine have questions about COVID, and this data and these samples are here for you to use. So I'm going to really talk about how we moved the identification of a gene called OAS1 to drug development within 12 months. And to do so, I'm going to talk about how can disease-influencing circulating proteins be identified without model systems. By model systems, we want to really just use humans to be able to identify causal circulating proteins. I'm going to talk about the genetic determinants of COVID-19 severity. That led us really to this protein called OAS1. I'm going to take you through what I think is a fascinating tour of the contribution of Neanderthals to this pandemic, how bats have influenced SARS-CoV-2, tool compounds that we're using to be able to influence pharmacologically the OAS1 pathway, and then we'll wrap it up with some questions or perhaps conclusions. So we really began this from a scientific point of view with asking a simple question. Can disease-influencing circulating proteins be identified without model systems? Why did we focus on this question? Really because circulating proteins are a favorable drug target, and also using some modern genetic epidemiology methods, we can identify with several assumptions causal circulating proteins that influence risk of disease. And when you do so, the probability of drug development success appears to increase importantly. And we do that by using a paradigm called Mendelian randomization. So Mendelian randomization is one of the favorite tools in our labs. And the reason why we use this is because we can estimate the effect of any exposure, in this case, we're going to talk about protein levels, on a disease without being biased by confounding. The way that we do this is we use genetic variants that are inherited in the population, and as you will remember from Mendel's second law, are randomly assorted prior to conception. And that random assortment is a bit like a randomized controlled trial, and it blocks the association with confounders and allows you to measure variation in an exposure like a protein level to then estimate the causal effect of that protein level upon the disease without this potential confounding factor. So no model systems relying only upon humans free living in their environments who can track human diseases, not models of disease, and you can imply causality without undertaking a randomized controlled trial. The big problem with these studies is that genetic variants could potentially influence the disease of outcome, acting on a pathway that is pleiotropic and therefore independent of the protein level. And when you do so, you could bias your results. So you could observe that a genetic variant influences the level of a protein, and you see the same genetic variant influences disease, and you could conclude that the protein is involved in the disease etiology, but it might be that that genetic variant is acting on a totally different pathway in the genome, as well as the protein level, and it's influencing the disease through that pathway. And if that were to occur, you could end up with biased estimates of causality. And so to overcome this, what we have done is use a very simple paradigm, and that is to use genetic variants that are right beside the gene that encodes the protein. We call these cis protein quantitative trait loci. You don't need to know that, but these are just genetic variants in or near the gene. Let's say they're in the promoter of the gene that influence the transcription or translation of that gene into a protein. And when you have such genetic determinants that are so proximal to the gene, it is very likely that they are influencing the protein level directly rather than influencing the disease through a pleiotropic pathway. And when that happens, you can rapidly observe causal effects of using human genetics without using any model systems. And when you can do that, then hopefully you can accelerate drug development opportunities. So in order to be able to apply such a paradigm, you need to be able to understand the genetic determinants of COVID-19 severity. And so very early on in the pandemic, we and other people around the world decided to gather all of the data that we could. So every country that was willing to contribute genome-wide association study data to a consortium, which then allowed us to rapidly identify several different regions in the human genome. These are all chromosomes. Each dot is a single nucleotide polymorphism, or just a base pair change in the genome. And the y-axis is negative log 10 p-value. And so what this allows you to see is the geography of the association of single nucleotide polymorphisms with the disease. And what this shows you is that this region of the genome has a very strong association with COVID-19 outcomes. And here you see one called OAS1, which has a less certain association with the outcome, but still achieves what's called genome-wide significance, which is above this p-value line here. And subsequent iterations of this program have allowed us to identify many other loci simply through increases in sample size, allowing us to fairly confidently predict that there is some causal mechanism that is influencing COVID-19 outcomes at these different loci. And I can tell you this was really surprising. So most infectious disease genetics has not been so successful in identifying loci which influence risk of disease through common genetic variation. And this definitely happened in the case of COVID-19. And so we said, aha, we've got an opportunity to try to dig into some of these signals using the Mendelian randomization paradigm, which brings us to OAS1. And so what we were able to identify is genetic variants that strongly influence OAS1 levels. OAS1 is both an intracellular and extracellular protein that allowed us then to apply the principles of Mendelian randomization to test whether or not the genetic variants that influence OAS1 also influence COVID-19 outcomes. And this is, again, to remind you, the circulating levels of OAS1. And we actually measured these in people who were uninfected. And so what this allowed us to do is say, okay, for people who've had a lifetime of higher OAS1 levels, would they be protected against severe COVID-19 outcomes? And I'm going to show you the results of this paper. What we found is that the non-infected OAS1 level, so the level that you have in a non-infected state, is strongly predicted to protect against severe COVID-19 outcomes. And so you can see the odds per standard deviation, per unit increase in the log exposure level of OAS1, that you decrease your susceptibility to COVID-19, you decrease the probability of hospitalization, importantly with odds ratio 0.68, with its confidence intervals described here, and consequently low p-values, to very strong protection against very severe disease, where very severe disease is defined as death or the use of respiratory support. Suggesting that increases, if you're a person who has a baseline increase in OAS1 levels, you would be strongly protected against going to the ICU. And what we then identified is that this is a protein, as an endocrinologist and being very naive, we of course didn't know what OAS1 did, but this is a protein that has actually been studied in infectious disease previously. And the genetic variant that we identified to be associated not with just OAS1 levels, but also severe COVID-19 outcomes is actually a splice site variant. So just to remind you, genes have different flavors when they're transcribed into proteins, and they can end up producing different isoforms. And those isoforms often have different exons in them. And that's exactly what's happening here, is this genetic variant is present right before the 6th exon of this gene. And what it does is it influences the presence of the 6th exon. And the people who are lucky enough to have this protective genetic variant G are the people who end up with the 6th exon. And so we're going to come back to that later, because colleagues at Glasgow have really dived into how exactly this P46 isoform protects against OAS1, against severe COVID-19 outcomes. And so this allowed us to predict that perhaps it actually was influencing not just expression of OAS1, but splicing of OAS1. And so we used, again, this Mendelian randomization paradigm to try to tease out the difference between splicing effects and expression effects. And what we saw is that the expression of OAS1 did protect, but quite mildly. And the confidence intervals for both the susceptibility and hospitalization estimate crossed the null. And when we restricted our analysis to predicting an effect of splicing of OAS1, and therefore the presence of the P46 isoform of OAS1, we saw very clear effects, such that there was a very strong protection against OAS1 if you were able to splice the OAS1 gene to contain the 6th exon. So we were able to observe that the signal, both for the OAS1 level and severe COVID-19 outcomes, really were coincident. This is called colocalization. It's a statistical methodology, which I won't get into, but allowed us to say with some confidence that this was the exact same signal that was driving both OAS1 levels, its splicing, and severe COVID-19 outcomes. And so we decided to take a deeper dive into this locus because, interestingly, this locus has a very fascinating history. So the OAS1 P46 isoform is present in individuals of sub-Saharan African descent. And we were able to use that information to basically deconstruct this locus to identify the causal SNP. And so this is looking at linkage disequilibrium. It's just a measure of how correlated all the genetic variants are across the region. And if you look in Europeans here in panel A, you can see there's a very high correlation across this whole locus. And so that's interesting because that doesn't really allow you to say which of these genetic variants is driving the signal, because this genetic variant could be just reflecting the effect of this genetic variant. But when you actually look at sub-Saharan Africans, you see that all of this gets disentangled rather beautifully, and that this single SNP stands out by itself. And if you were able to test the effect of this SNP upon severe COVID-19 outcomes in African Americans, you could then provide some insights as to whether or not that is the causal SNP. And so we did this work with Jenny Huffman, who's at the Million Veterans Program in the US, who's very helpful in providing this data point here, which is really salient because it provided 1,670 African-American cases with severe COVID-19 outcomes and over 100,000 controls. And what we were able to do is show that the summarized African-American effect at this single genetic variant was strong, was protective, and almost exactly the effect that we saw in Europeans, suggesting that it was not another genetic variant at this locus, but this was likely to be the causal SNP itself. And so this gave us some confidence that OAS1 was actually the causal signal at this locus, because this SNP is the one that controls the isoform of OAS1. And so we naturally asked the question, what does OAS1 do? Well, when we asked the virologists, they laughed because they said, oh, you geneticists, you found something we've been working on for years, and that's exactly the case. So what OAS1 does is it actually leads to the activation of an enzyme called RNase L. And RNase L is like a pair of scissors that runs around inside your cell and chews up double-stranded DNA, double-stranded RNA. And so if you have a virus that's replicating inside your cell, the RNase L system is the one that degrades that double-stranded RNA. And so it's a host protective mechanism that allows you to prevent replication of things like SARS-CoV-2, which are double-stranded RNA viruses. And interestingly, there are two drugs that have been worked upon which actually increase the activity of RNase L by inhibiting OAS1. And these are PDE12 inhibitors, as well as interferon beta 1b, which I will describe in a few moments. And so often in the course of trying to understand a really complicated system like predisposition to severe COVID-19 outcomes, it's helpful to look at data from different points of view. Because you could look at things from a human genetics point of view and come to one idea about what's happening. But if you look at using a different set of methods, you may come to a very different understanding of the outcome. And so to do that, what we did is we looked at the effects of measured OAS1 levels in the BQC19 cohort. And we found exactly what we had predicted from the genetics, is that the people who in an uninfected state had higher OAS1 levels, and particularly the P46 isoform of OAS1, were strongly protected against severe COVID-19 outcomes. Okay, so that gives you hopefully some understanding of how we got to trying to dive into OAS1, our rationale for doing so. And now I'm going to take you through what I think is the coolest part of the talk, which is really how this came to be in humans, that we have this very strong protective factor against OAS1, against SARS-CoV-2 infection. Okay, so let's just go over a little bit of Homo sapien history, which I'm sure you're all aware of, but in case you've forgotten, Homo sapiens, of course, arose out of the Rift Valley in Africa. The lazier ones, like my ancestors, stopped in Europe, and the more adventurous ones went all the way across Eurasia to the tip of Tierra del Fuego. And what happened is when Neanderthals left, as you remember, Neanderthals left a lot earlier, but at least 60,000 years earlier than modern Homo sapiens, and they generally settled in Northern Europe. And when they left sub-Saharan Africa, they took with them, as you would expect, the P46 isoform. And that tells us that humans have been under pandemics for thousands of years, and that they took this with them because it protected and was selected for to prevent the outcomes of RNA, double-stranded RNA viral infections such as coronavirus pandemics. But what happened when the modern Homo sapiens left the Rift Valley is they lost this P46 isoform, this very crucial tool that protects against coronavirus infections and other double-stranded RNA infections. They lost this P46 isoform, and we don't know why, and we don't know how. But thankfully, what happened is that those early Homo sapiens who were Eurasians at the time met with Neanderthals in Northern Europe, and they interbred with these Neanderthals who reintroduced the P46 isoform into the population. And you may be wondering why I'm showing this gentleman here. This is Hugo Zeberg, who's been a wonderful collaborator through this whole pandemic work that we've done, because he is actually a paleogeneticist and understands all of the methods that are required to be able to understand the contribution of archaic DNA, such as Neanderthal, to the prevention of common disease outcomes, such as COVID-19. I'd also add as a side note that he is also an emerge doc, making him the only emergency physician that I know who's also a paleogeneticist. So let's get back to the Neanderthals. Okay, so now the Neanderthals give back to the Eurasians this protective factor, and then an explosion happens. The frequency of that allele goes from about 5% of the population to about 32% in modern Eurasians. And this is work that was done by Hugo, published in PNAS earlier this year, demonstrating that there is a very important change in the allele frequency in the population, likely due to positive selection upon this allele to ensure that humans are protected against double-stranded RNA viruses. And we don't know exactly what they were protected against, but this strongly suggests that our ancestors have undergone many pandemics, which has allowed this allele frequency to increase. Now, if this allele was able to protect against many diseases, it would actually go to 100% of the population. But that's not what happened at all. What you can see is that it actually ends up being about 30% of individuals carry the allele across all of the Eurasian populations, and then it just stops there. It stops increasing in frequency. We have no idea why, but this probably suggests that there's some balancing selection going on, decreasing the frequency of this allele in the population because it's protecting, because it has some other deleterious effect. That's a suggestion. We provide no evidence, but it's possible that that's true. And so I'm now going to talk about bats and how we've learned from bats to understand this OAS1 protein. And to do so, I'm going to show you some data that was published recently at the end or about the middle of 2021 from a lab in Glasgow from somebody who's become a close collaborator. Now his name's Sam Wilson. So Sam's actually a virologist. He understands viruses unlike us. And what he was doing at the time, unbeknownst to us, was really diving into OAS1 because he had added interferon to cellular systems to observe what are the interferon stimulated genes, because doing so would hopefully allow you to identify some of the protective factors for COVID-19 outcomes, because of course our body uses the interferon system to stimulate our immune system to protect against the SARS-CoV-2 virus. And what Sam and his colleagues were able to show is this is actually a measure of replication of SARS-CoV-2. And this is a control. So this is just a vector. And you can see that this is what amount of replication that you would expect, to which Sam and his colleagues add P46. So this is the isoform that I told you about of OAS1. And when you add that, you see a great decrease in the ability of SARS-CoV-2 to replicate. This is the other isoform of OAS1. So this is the flavor of OAS1 that many people in the population have that actually appears in this model system that have no effect upon SARS-CoV-2 replication. And so I told you that I was going to talk to you about bats. And that is because Sam brought some really interesting data and hypotheses to the table. So what Sam was able to point out in his colleagues is that we're surrounded by coronaviruses. We've had lots of coronavirus exposure in the past as a species, and we're not the only mammals that are exposed to the coronaviruses. So why is it that OAS1 is particularly helpful against protection from SARS-CoV-2? So what he was able to demonstrate is that in the MERS virus as well as OC43, which are both double-stranded RNA coronaviruses, these viruses actually evolved within a species that has the P46 isoform of OAS1. So not just humans have this P46 isoform, but other mammals do too. And interestingly, these viruses have evolved under the pressure of the OAS1 system. And the way that they have fought against those mammals to stay alive is that those viruses have made an inhibitor of OAS1, and that inhibitor is phosphodiesterase. And so natural selection across different species has repeatedly shown that the way that double-stranded RNA coronaviruses like to fight against the OAS1 system is by generating PDE, which then actually acts to decrease the activity of OAS1, allowing them to replicate within these species. But what's happened in SARS-CoV-2, should it have come, and we don't know exactly where it's come from, but leading candidates are pangolins, as well as this subspecies of bat called Rhinolohidia. And Rhinolohidia and pangolin don't have the P46 isoform of OAS1. And SARS-CoV-2 has not had to fight against the P46 isoform of OAS1, and therefore never developed this phosphodiesterase system to be able to decrease the activity of OAS1. And that means that this is one of the Achilles heels of the SARS-CoV-2 virus, because it's exquisitely sensitive to the P46 isoform of OAS1 because it's never had to fight against it before. And he shows this here by showing the effect of the P46 isoform from Homo sapiens on replication of SARS-CoV-2. This is what I've already shown you. It decreases tremendously. But when you use the OAS1 isoform from the species of bat, I'm sorry, I can't pronounce it, but Rhinolohidia, I think is what its name is. It has no effect upon SARS-CoV-2 replication. So this suggests that pharmacological interventions that act upon in exaggerating our effect of OAS1 or exaggerating increasing OAS1 levels could protect against SARS-CoV-2 replication and therefore the severity of disease. So I promised that I would be brief, and I know that you're all very busy, and so I'm just going to finish with some slides here to just review what's essentially happened since we began working on this pandemic. Is that through work that was led by Andrea Gana, we started out knowing nothing about the genetic susceptibility to SARS-CoV-2, but really through international collaboration, we were able to identify multiple loci that influence SARS-CoV-2 severity in the human population. We were able to boil down one of these areas of the human genome to a single likely causal SNP. This allowed us to look at the splicing effects of this SNP, leading us to suggest that P46 isoform was influencing the severity of SARS-CoV-2, which led us to be able to measure the protein levels of OAS1, showing a clear and protective effect of OAS1 levels on SARS-CoV-2 outcomes, which allowed us to undertake a Mendelian randomization experiment, observational epidemiology, as well as in vitro validation, which was published last year by Sam Wilson's group in Science. And so what we've been working on very fervently with collaborators within the industrial space is using tool compounds that actually inhibit phosphodiesterase 12 so that we can test the effect of such medicines upon replication of the SARS-CoV-2 virus within a model system, so that our drug development partners can justify the investment that it would take to be able to develop a medicine which would protect against double-stranded RNA viruses. And I just said double-stranded RNA viruses and not SARS-CoV-2 because the drug development offered opportunities for enhancing the host against SARS-CoV-2 infection are probably going to become increasingly limited as other medicines come to market. But what appears to be the case for inhibition of PDE12 and increasing OAS1 levels is that this would not be helpful just against SARS-CoV-2 infections, but other double-stranded RNA viruses, which has brought to the table a lot of interest from our pharmaceutical colleagues who are working on development of such compounds now. So to summarize, increased OAS1 levels protect against COVID-19 susceptibility, hospitalization, death, and ventilation. These analyses survived multiple sensitivity tests, which I'd be happy to describe. OAS1 has clear biological rationale, which was obvious to everybody except me prior to these experiments. Natural selection points to phosphodiesterase is the key enzyme to inactivate OAS1, and so giving a medicine that inactivates PDE would lead to increased activity of OAS1. Compounds exist to increase OAS1 levels, and we're currently testing them with our collaborators, particularly Sam Wilson and a woman named Anna-Sara Lehmann in northern Sweden, to test for their inhibition of SARS-CoV-2 replication. Actually, going to take a little bit of time to thank people. So the BQC19 was a fairly exhausting effort for which many people across McGill put a lot of time into, and I'm just going to take a moment to talk through this because it's important. So Vincent Musser really led the entire program, brought a lot of money to the table so that we could collect data and samples, which are available to you to use, or anybody can use for their own studies. Daniel Kaufman has been a very close collaborator in this. He's actually an infectious disease specialist. He's actually a virologist. And so he was very helpful in helping us to understand the OAS1 system so that we could try to provide some insights to the community. All of this really only happened because we got fast money. And we got that really quickly from Lawrence Rosenberg, Rod McKittis and Jerry Batches, the leaders here at the Jewish General Hospital, who allowed us to spend money really without consequence to collect data and samples as quickly as we could. We had about 2 weeks to put together the entire biobank. Usually we put this together for about 2 years, and they just let us spend money with reckless abandon, and that enabled us to be able to launch things quickly. David Morrison, Mark Aflalo, and Jonathan Aflalo really led the collection of the samples and data here. With people who you will remember, but in the spring of 2020, we didn't have enough PPE for these people. So PPE were restricted only to clinicians. These were researchers who were hanging out in the emergency room, talking as little as possible to COVID-19 patients, but were definitely repeatedly exposed at a time when we were told that there was physicians being ventilated en masse in Italy. These people volunteered. They were most of them were recently laid off from research programs which had been canceled due to the pandemic. They volunteered to really put themselves in the line of fire with no PPE to collect these data and samples. So really a big thank you to all of these people. Mark Lathrop and Yanis Vergusi and Danielle, Guillaume Bourque, and Pierre Lepage allowed us to both sequence and genome-wide genotype these people. The administrative people who really pushed through hiring a team of 20 people within a week, which caused them to have a lot of late nights and a lot of administrative work, which usually takes months to be done in days. All of the ethics was really done by Man Zawati as well as Jan Jali, and we were very lucky that a lot of this work was done prior to the pandemic, and so we were able to have ethical approval quite quickly, but that was done really through leadership from Kathleen Blagrave as well as Bessie Bitsas. Vince Rogetta and Celia Greenwood really led a lot of this analysis. I can't tell you how important it is to have an interactive legal team, which we're very fortunate to have here at the Jewish General Hospital. We've moved data and samples from the Jewish to 32 groups around Canada, and this has only happened because they've accelerated this program. We needed to process these samples. We didn't have a wet lab to do that, which could handle the virus, and so Erwin Schur and his team, as well as Bloomer Brenner and her team, Maureen and Nathan, collected these samples and processed them with Don Vin. And then we had an IT team collecting all of the data here at the Jewish. We received money from Bram Friedman, the foundation. Sophie Desmarais wrote us a check very early on in the pandemic, which allowed us to get to work quickly. And then the bulk of the funding came from PHAC, FRQS, and Genome Quebec. I'll stop there and thank our funding partners again, and be very happy to have any questions, concerns, or comments that anybody has.
Brent, thank you so much. Really outstanding lecture. Really a pleasure to listen to this. Congratulations to you and the team for all the work that you have done. George, you're there. George and I are fielding questions. George, do you want to get started?
Sure. There are a few in the chat that maybe we can start with. One is from David Rosenblatt here. What about LZTFF1? Was that already studied?
Excellent question, David. Yeah, so we've been working on that locus for, definitely not 2 years, but a year and a half. There are lots of genes at this locus. We have no idea what's going on. There has been a high impact paper published on every gene at that locus, all claiming that they have separately identified a different causal gene. I don't know what's happening there. If you know me very well, I don't really like to work on complicated problems. We like to work on simple problems. We haven't worked very hard on the chromosome 3 locus or the LZTFF1 locus, just because it's so incredibly complicated. And the reason why it's so incredibly complicated is due to the Neanderthals. So it's actually, what I showed you is a protective genetic variant that was given to Eurasians by Neanderthals. What's happening at chromosome 3 is that there's a very strongly deleterious variant at chromosome 3, which is given to us by Neanderthals. And when that happens, because Neanderthals so recently introduced their DNA to us about 60,000 years ago, the linkage disequilibrium block is really long. And when that happens, it's really hard to disentangle what the causal gene is there. So excellent question, top of everybody's mind in the field, and we don't have a good answer.
Thanks. Dr. Sami is asking if the mRNA vaccines affect the levels of the OAS1.
We don't know. So we actually have samples that we are trying to answer that question. We don't probably have enough samples to be able to answer that question yet, but we would suspect that it would, and that's because the mRNA vaccines actually act in part by stimulating the interferon system, and the stimulation of the interferon system leads to increased OAS1 levels. I strongly don't think that this is why the vaccines are affording protection, because of course the OAS1 levels would come back to normal after a few weeks or even a few days. But what is very clear is that those vaccines last for months at a minimum. And so while we would expect OAS1 to increase in the acute phase, it's almost certainly not having a protective effect long-term in people who've been vaccinated.
Another question from David. What about levels in the animal vectors, mink, deer, and other species?
Yeah, so it's a good question from David. So a lot of these animals have not been looked at very carefully. Most animals have a P46 isoform of OAS1. It's fairly conserved across mammals, but as Sam Wilson pointed out, in the 2 species from which the SARS-CoV-2 virus has likely jumped to humans, there's no P46 isoform, which may be why it's so exquisitely sensitive to the P46 isoform of OAS1.
Another question from Dr. Klein. How do you reconcile the evolutionary genetics with the global epidemiology in which Northern European countries and those populated with their descendants have had the highest rate of infection and mortality, whereas African countries have been remarkably spared?
Yeah, okay, so let's not make too strong conclusions here, because while ancestry is important, there's also very clear effects, social effects upon exposure to virus, who's working in high risk professions, who are frontline workers. So, it's always important to couch any evolutionary arguments in the context of society. With that as a caveat, what we do know very clearly is this huge risk factor that's very common on chromosome 3, which is the one David brought up, LZTFF1. We and others have shown that if you're under the age of 60, your probability of death, if you have that genetic variant, increases about 3-fold. We published that in PNAS. That was Tomoko Nakanishi who published that. And so that's probably contributing to some of the differences that we see across different countries. But again, I'm going to come back and be cautious and say that there's lots of differences that are not only genetic between, for example, Northern Europe and Africa and Sub-Saharan Africa.
So regarding the phosphodiesterase sort of information, Dr. Sami's asking about Viagra. Is it going to be?
Yeah, no, I mean, a natural question, and it's like very, very good. And so we actually sort of dove into that. So Viagra doesn't specifically inhibit PDE12 very well. So we actually partnered with a company who's been very helpful. It's a company called Cyclica RX, and they're a medicinal chemistry. They said very early on in the game, it's very unlikely that Viagra is going to work because it doesn't actually inhibit phosphodiesterase 12. Phosphodiesterase 12 is the enzyme that inhibits OAS1. And so we wouldn't predict that Viagra would work, and we have no evidence that it does. It's really specific PDE12 inhibitors. And so that's why we're working with our pharmaceutical partners to be able to focus on PDE12 inhibitors. Would have been great if, and it would have been a great story if Viagra had worked, but unfortunately that's not the case.
So let me ask a question to clear up my understanding regarding that area of phosphodiesterase. What about the people who don't have P46 because of the splicing difference? Would a phosphodiesterase inhibitor do anything for them?
Lovely question. So one that we've been struggling with since we really published this paper. So we've been trying to look at that question if there are a few different lenses, George. One of them is that you can give a PDE12 inhibitor to a cell line that doesn't have the P46 isoform. So you take a cell line, you could, optimally it could be a long alveolar cell line. You could give it a SARS-CoV-2 infection, and then if it didn't carry the P46 isoform, that would allow you to experimentally test whether or not PDE12 inhibition was helpful in those people. So we're actually doing those experiments now. The thing that's been remarkable to me, I didn't, of course, really work very much in cell lines. I didn't really know that a lot of cell lines, nobody's ever sequenced them. So some of the cell lines, we've actually had to go back and sequence them and find out if they carry the genotype, which would afford the cells the P46, which was shocking. Nobody's ever sequenced these cells, but anyways. So that's one of the things that we've had to do. We have a cell line that Sam Wilson is working on that is a P46 heterozygote, and will allow us to be able to test the effect of PDE12 inhibition on increasing OAS1 levels through the P46 isoform. But what we're trying to do is we're trying to see if people who do not carry any of the P46 alleles, so they actually don't make any P46, we're trying to see when we measure RNA expression of their OAS1. So it should be a fairly ineffective OAS1. We're trying to see if RNA expression that's increased in people, because there's other determinants of RNA expression of OAS1 other than this one genetic variant, if more of the, let's say, lousy OAS1 is helpful at all, because that would allow us to predict if the drug would be helpful in those people.
Right.
We're doing those experiments now. The RNA sequencing has been dreadfully slow, and Tomoko Nakanishi is actually working on that question. So we don't have any results yet, but hopefully we would have some. But it's an excellent question because that would allow us to be able to predict, would a drug be helpful in the general population, or would you actually have to genotype the people first to only give it to the people who have the very effective form of OAS1, which is P46?
Or it could be non-specific effect, some other.
It could be a non-specific effect, yeah. So we don't know yet, but the real, drug development is a slow business, and it's amazing how fast it's gone in COVID. But we hope to have an answer, at least from an RNA level, in the next few months.
That's great. We have one other question. Considering that SARS-CoV-2 originated from the Wuhan province in China, are there data indicating an abnormal allele frequency of P46 in that area, above or below the 30%?
Good question. No, my answer is no, but I don't know the answer to the question. But, if you just look here, can you see this one? Yeah. So if you just look, I wouldn't suspect so, Will. It's possible, but you can see that the allele frequency is very stable across Asia. Is it possible that the Wuhan province has a different allele frequency? Yes, and I provide no data to suggest that it is not possible, but I think it's improbable, just because everybody around it, whether or not you're in Japan or in Thailand, has little frequencies that are reasonably stable around 30%.
Yeah, I was. I'm sorry if I'm allowed to unmute.
Yes,
but I was just.
go ahead.
Yeah, I was. I'm Will. I'm the one who asked the question. I was just wondering because you showed that the species of bats in the pangolin had an absence of the P46 isoform. So I was wondering if it just so happened to fall in an area of the human population that also had a decrease in that isoform as well. But
it's a good idea. It's a good idea because you would expect then that those people would be more infectious and it would be more transmissible. And it's possible. I just don't personally have any data from the Wuhan province. Yeah.
well, regardless, I really enjoyed this talk. Thank you.
Thanks.
Pleasure.
Are there any other
Yeah, go ahead, George. I
questions?
don't
Any
questions
other
as
questions?
close out the meeting. Any other questions from anyone? You have 30 seconds. Brent, once again, really outstanding, truly outstanding lecture, and needless to say, outstanding work on you and your team, because I know it's a many people involved in making this and getting this data and making this happen. Really, congratulations and thank you for joining us at Medical Grand Rounds. Thanks, George, for fielding questions today. And on that note, we will end
Thanks,
round. Thanks everyone for participating. Have a great day.
Thanks for having me. Bye now.
Bye-bye.